boys. Last time we see structure of typical flower in detail. Today we learn development of male gametophyte. Development of male gametophyte start from pollen grain. So pollen grains are called first cell of development of male gametophyte. Development of male gametophyte, it complete at two different site or two different places. Before pollination in anther and after pollination on stigma. We see these both the steps or stage one by one. Before pollination, it's a androecium. It is male reproductive whorl. It made up of two parts. This uppermost part called anther, and this stalk-like structure it called filament. Anther is of two types: monoecious anther and dioecious anther. Monoecious means single lobed and dioecious means bilobed. Each lobe consists two pollen sacs. Monoecious anther have two pollen sacs and dioecious anther it have four pollen sacs like this. Each pollen sac contain MMC means microspore mother cell. It also called pollen grain mother cell. The nature of pollen grain mother cell is diploid. These diploid pollen grain mother cell undergoes meiosis and each pollen grain mother cell produces four haploid pollen grains. After formation of haploid pollen grain, these pollen grain again divide by mitosis and forms two-celled stage. When pollen grains are present in two-celled stage, anther undergoes dehydration, means dehiscence, and it get broken down, ruptured down, and pollen grains comes outside. You know boys, sometimes pollen grains are comes out in two-celled stage, and sometimes it may be comes outside in three-celled stage. These pollen grains look like this structure. Pollen grains are first cell of development of male gametophyte or male gametes. These pollen grains are unicellular, uninucleate, haploid. They are light in weight, sticky, and look like dust particle. Each pollen grain have two layers. Uppermost layer called enzyme, inner layer called entype. This upper exine layer, it have ciliated structure. These ciliated structure called spines. This exine, it made up of chemical sporulae. It is thick and protective in function. On exine, microscopic openings are present. These microscopic openings called germ pores. Number of germ pores are different. Monopod pollen grain have single germ pore and dipod pollen grain possesses three different germ pores. These germ pores act as a site to exit the pollen tube. It's a two-celled stage of pollen grain. Generative cell and vegetative cell. Vegetative cell and generative cell, they have difference. Generative cell is small, but it have large nucleus, and vegetative cell, it looks large but have small nucleus. Vegetative cell, it have reserve of food material. Generative cell, it do not have reserve of food material. Vegetative cell, it called a tube cell, and generative cell, it called sperm cell. Here, first stage is complete before pollination. We turn to second stage after pollination. Pollen grains are light in weight, small, sticky, so it easily transfers from one place to another place by insect, birds, animal, wind, water, and so on, and dust on stigma. It's a female reproductive whorl, gynoecium. Gynoecium, it also called carpel. It may be known by the name pistil. It is made up of three parts. This lowermost solan part, it called ovary. This middle tube-like structure, it called style, and this uppermost structure called stigma. Pollen grain dust on stigma. When pollen grain dust on stigma, they start to match the receptor with the receptors of stigma. When receptors of pollen grain and stigma match with each other, then germination begins. This process is similar to key and lock. If receptors of pollen and stigma not match, stigma does not allow pollen grain for germination. After matching the receptor, stigma produce sugary alkaline liquid. Pollen grain absorb sugary alkaline liquid. It absorbs more and more sugary alkaline fluid, and due to which internal pressure of this entire get increases, and this entire wall it break any one germ pore and comes outside in the form of tube like structure which we call pollen tube. After formation of pollen tube, first tube nucleus enters, then cytoplasm enters in pollen tube, then generative nucleus enters, and this generative nucleus is divided by mitosis to form two male gametes. It forms two male gametes. This three-cellular stage called development of male gametophyte. Before formation of two male gametes, at the neck of pollen tube, a lock create, it called callus plug, and this callus plug it prevent backward flow of cytoplasm in entire wall. This overall process called development of male gametophyte. Now we stop this point here, and remaining point we see in next video. Boys, if you want to see the next video, then subscribe the channel and press bell button. Thank you.